We just got done talking about the B cells, or the B lymphocytes, and how they have antibodies on the surface that recognize an antigen that's floating around. But you might have thought, what happens if the antigen, the dangerous antigen, is not just floating around in the fluid? What happens if it's inside one of our own cells, for example, a virus? In that case, the B cell can't do anything. B cells only work in humoral immunity in the fluid. We need another kind of immune cell to take care of antigens that have gotten inside our own cells, and that is the T cell. The first kind of T cell we're going to look at is the killer T cell, and if your body's a castle, this is the executioner. It's also called a cytotoxic T cell, and the role of this cell is to identify and kill all the native cells that have been infected by a virus. So this cell needs some way of knowing, some way of detecting which of the body's cells are healthy and which have been infected by a virus and need to be killed for the sake of the rest of the body. When a cell is infected by a virus, the virus inserts its genetic information, be it DNA or RNA, into the host cell and uses the host cell's machinery to make these abnormal peptides. They're abnormal because they are not typical to be found in our bodies. And the cell, what it does is it puts these abnormal peptides through the endoplasmic reticulum and it inserts them in this little display protein called a MHC Class 1. It shuttles these two through the Golgi into a vesicle and ultimately back to the surface of the cell. So that U shape is the MHC Class 1, and the viral antigen, the one that came from the virus, is in red. This cell is showing everyone around it, hey, this is what's inside me. It's showing there's a viral antigen inside me. It's like a cry for help. And the killer T cell is the one that's going to respond to that cry for help. When the killer T cell, or here it's called cytotoxic, when it recognizes that antigen, which is in orange, displayed on MHC Class 1, the cytotoxic T cell will activate. Now, important to note that this particular killer T cell will only detect this triangle orange antigen. There are other killer T cells to detect all the other kinds of antigens there could be, but this one is specific to this antigen. When a killer T cell becomes activated, that means it has recognized, okay, this is a human cell, our cell, but it's been infected by a virus. It will release factors to induce cell death in the infected cell. So this is essentially saying, okay, I know you're one of our own body's cells, but you're infected by a virus. I'm really sorry. We have to destroy you so that the virus is not allowed to replicate. So it releases these factors, causing destruction of the infected cell. The job of the executioner is not easy. There's another kind of T cell that I've saved for last because it's the helper T cell, and it's essentially the supervisor of all the other ones. So if the B cells were the guards, the killer T cells were the executioners, the helper T cell is supervising all of them. Its role is to check on all the other immune cells and check whether they've identified their antigens correctly. And if they have done that correctly, then the helper T cell will instruct that other immune cell on how to respond. So the helper T cell is always interacting with a different immune cell. Here it's labeled as an antigen-presenting cell because remember that cells can, if they have encountered an antigen, they can display it on their surface showing, hey, here's what I found. And essentially these cells are going to their supervisor, hey, this is what I found. Is it right? Am I right that this is an antigen that should be responded to? This interaction between various membrane receptors is what mediates the job of the helper T cell. We have an interaction between CD4 on the helper T cell and MHC Class 2 on the antigen presenting cell. These interactions are going to trigger the release of factors from the helper T cell as well as from the antigen presenting cell. Signals that are essentially confirming, yes, we've found an antigen correctly and we need to take action. So I know all along I've been saying that each of these cells activates, but most immune cells, they will not activate unless they are told by a helper T cell to activate. So let's take our first example, which is the killer T cell. This is what I showed you before. The killer T cell will release factors telling the virally infected cell to destroy itself. It needs a signal from a helper T cell in order to proceed with that. It needs the thumbs up from its supervisor before it does that. And what I mean by thumbs up is the release of factors from the helper T cell to the killer T cell saying, yep, you got it right, go ahead and release your factors. A T helper cell will also help a B cell. Here we have a B cell, remember those involved in humoral immunity, and it has detected these blue square antigens. A B cell will also internalize some of these blue square antigens and display them on its surface as if to say, hey, this is what I found. Telling its supervisor, hey, this is what I found. Should I really react to this? And when it displays it, the helper T cell that is specific to this antigen will perform this receptor interaction, and the helper T cell will release factors telling the B cell, yes, go ahead and activate. When the B cell activates, it will make many plasma B cells to create antibodies, as well as memory B cells that are not shown, unfortunately. The helper T cell also mediates the actions of macrophages. So these macrophages, these are the ones involved in innate immunity. They'll just eat any foreign cell, they're not specific. But a helper T cell, when it activates a macrophage, will speed up the actions of a macrophage. It will make the macrophage perform phagocytosis more efficiently. So in summation, the adaptive immune system, we can think of it as two branches: the humoral immunity, which is taking care of pathogens in the fluids of the cell, and that's taken care of by the B cells. And then the cell-mediated immunity, which is done by T cells, the cytotoxic or killer T cells, which destroy cells that have been infected by a virus, and the helper T cells, which are like the supervisor. They confirm and they mediate the actions of the other immune cells based on their detection of that specific antigen. So here we go back to this primary and secondary immune response graph. On the y-axis is our antibody concentration, and that's our measure of how well the body is responding to the infection. There's antigen A, and so that's a specific disease. Say you're exposed to antigen A for the first time. Your body will produce some antibodies to fight off this infection, but it will produce them slowly, and the maximum amount it produces is not very high. That's because there are no memory B cells yet. T cells also produce memory versions of themselves when activated, so that's why this says no memory cells. There are no memory B cells or memory T cells. So essentially the body has very few cells around that will be able to respond to this particular antigen. Some time passes, and the person is exposed to antigen A again, and then a second disease, antigen B. And what happens is that its response to antigen A is to make very quickly a lot of antibodies against this antigen. And for antigen B, since it's the first time it's been encountered, it still produces that slow and not very large response to antigen B. The key here is that the second exposure always elicits a stronger, greater, and faster immune response than the first exposure, because at the time of the second exposure, there are memory B cells and memory T cells, more of them than before, that are ready to respond by producing antibodies and releasing the factors that are necessary to fight off the infection. Our knowledge about the adaptive immune system has helped us to develop vaccines and eventually eradicated certain diseases in the world. A vaccine consists of small chopped up pieces or inactivated pieces of a molecule that would otherwise cause disease. For example, the influenza vaccine consists of small inactivated pieces of the influenza virus. When that is injected into a person, the pieces don't cause any symptoms of a disease, but they do look enough like the original disease that they elicit an immune response. They are bound to by antibodies, and the B cells that are specific to this antigen are activated. Those B cells will produce memory B cells of the same kind. And that is important because the memory B cells will stick around for a long time. Down the line, when this person gets infected with the actual disease, the one that will cause symptoms and perhaps very dangerous, this person has memory B cells because they were exposed to the vaccine. The memory B cells, there will be lots of them, and they will readily produce antibodies quickly, and a large amount of antibodies, in order to cover up these antigens, have them all phagocytosed, destroyed, and the body will successfully fight off this disease. Whereas somebody who hadn't been vaccinated, they would not have the memory B cells, they wouldn't produce enough antibodies fast enough, and their body would not be able to fight off the disease as effectively. Now, to finish off, I want to talk about a couple of cases where the immune system malfunctions and what kind of conditions that causes. David Vedder was a boy known as the boy in the bubble. He had a severe combined immunodeficiency, which meant that his T and B cells were both defective. They were not able to recognize the antigens that they should have recognized. When he was born, he had to be placed in a sterile plastic bubble, and he only ever interacted with people through this bubble and through like gloves sticking out of the bubble. This was because even the slightest antigen that he was exposed to could easily kill him. He had no immune system, so the common cold would mean surely a death sentence for him. When he was 12, doctors proposed that they would give him a bone marrow transplant from his sister. T and B cells both originate in the bone marrow, so this seemed like it would be a good solution. They screened his sister, her bone marrow, and they tried to remove all the bacteria and viruses that were present. They found she was a good match. They did the bone marrow transplant, and unfortunately, a week later, he became very ill and died. When he died, he had hundreds of small tumors in his body because his sister's bone marrow contained a single virus, an Epstein-Barr virus, that was not hurting her at all, but for him it was deadly because he had no means of defending against it. Fortunately, a couple years after his death, the technologies improved for bone marrow transplants, and now children born with SCID don't have to live in a bubble. A much more common disease of the immune system is HIV. HIV is a virus which invades the T cells, the very cells that should be protecting your body from viruses. And people living with HIV need to get blood tests to measure their T cell count. When their T cells are high, that means the virus is latent or dormant. It means their genes are present in the T cells, but they aren't making new copies of the virus, so the T cells are still working fine. When the T cell count is low, this indicates that the virus has begun assembling copies of itself and destroying its host cells. At this stage, the person is diagnosed with having AIDS, and due to their lack of T cells, they are immunocompromised. This means they're highly vulnerable to opportunistic infections. So when a person with AIDS dies, it's usually due to an infection like pneumonia or thrush that a healthy person would have had no trouble fighting off. So this example can teach us a lot about the nature of the immune system and the role of T cells in adaptive immunity. Thanks for watching.